When we ask ChatGPT about the current most advanced way of cancer treatment, the first thing we retrieve is immunotherapy and targeted therapy. The cells of the immune system are continuously monitoring our tissues. Dendritic cells activate cytotoxic T cells, which can then sense tumor-associated antigens, using their T cell receptor and other coreceptors. Once activated, CTLs release perforin and granzymes, causing them to die by apoptosis. However, due to the presence of an immunosuppressive microenvironment, tumors are not recognized and eliminated by the immune system. Some tumor cells express inhibitory molecules such as PD-L1, which binds to PD-1 receptor on CTLs and deactivates them. Meanwhile, tumor cells can attract immunosuppressive cells. The most important ones are protumoral M2-type tumor-associated macrophages. Therefore, it is a great challenge to conduct targeted and on-demand modulations on the tumor immune microenvironment to maximize the immune effect. Based on the above background, the research team of Yiyao Liu and Chunhui Wu, at the University of Electronic Science and Technology of China, published an article in APSB, entitled Biomimetic Gemini Nanoimmunoregulators Orchestrated for Boosted Photoimmunotherapy by Spatiotemporally Modulating PD-L1 and Tumor-Associated Macrophages. The Gemini nanoimmunoregulators were constructed respectively by loading PD-L1 inhibitor metformin and toll-like receptor 7/8 agonist resiquimod into the highly biocompatible mesoporous polydopamine. Subsequently, they were individually wrapped with peptide T7 and M2 engineered red blood cell membranes for specific targeting capability of cancer cells and TAMs. First, the PM@RM T7 nanoimmunoregulators showed excellent tumor cell-targeted endocytosis via T7 transferrin interaction, triggering photothermal ICD phenomena, and PD-L1 reduction at the cellular level. These data demonstrated that PM@RM T7 functions as a targeted ICD inducer and an immune checkpoint blocker. The other, Gemini nanoimmunoregulator, PR@RM M2, has an excellent ability to target M2-type macrophages by M2 peptide. It effectively repolarized the TAM's phenotype to M1 type. Enhanced phagocytosis acted as antigen-presenting cells to activate CTLs and released more pro-inflammatory factors to inhibit tumor growth. Furthermore, Gemini nanoimmunoregulators, G6 group, showed significant tumor suppression towards the bilateral breast cancer model compared with the single immunoregulator. By analyzing the ratio of immune cells such as DCs, CTLs and TAMs, it was found that Gemini nanoimmunomodulator successfully remodeled the tumor immune microenvironment. Also, they systemically and effectively inhibited tumor cells' lung metastasis. In summary, we constructed Gemini nanoimmunoregulators that spatiotemporally modulate tumor cells and macrophages in the immune cycle. It not only activates the immune response but also revitalizes the immune microenvironment, providing a reference for the next generation of precision immunotherapies.